So, Divya?
Takes a minute. Just I just close this. Yeah. It's not coming. I have the Mac connector. Yeah, it's Mac connector. I think maybe that point. Oh, it's VGA.
Yeah.
Yeah. Yeah, pointer I will use. I will use the key. That's fine. That's fine. But just for pointing, I can use. No? Okay. So it is coming like this. So if I do the other way around, then it goes in the meeting, it goes, can you see in the meeting, it goes the other way around, right? It's fine in the meeting now? Oh, okay. This is the problem. It's coming fine here. No, it's showing first now.
Stakeholders. Then we can go for it. Elizabeth space. Yeah. It will simply arrangement. Yeah. Yeah, middle.
Okay.
Okay.
Yes, great. Thanks. Good morning, everyone, and sorry for the technical glitch. So first of all, I would like to thank Professor Ashwini, Professor Niranjan, and Professor Pal for inviting me here. This is my first visit in ISER, Kolkata, and these are some of the pictures I just took yesterday. And I'm really happy to be back, as this conference has a theme of collecting the theoreticians who are in Kolkata region. So I would call myself pseudo Kolkata person, because I had the privilege to be in the theoretical soil for 2 years when I was faculty in IIT Kharagpur before I moved to IIT Delhi. So I'm very happy to be here and I welcome you all to my talk. But before I begin, um, yeah, this is also beginning of the in-person meetings now we are having, and we are now visiting places and meeting people again. Feels nice, but let's just step back 2 years back. The situation was not so good, and some of us are new faculty here, and we are called like COVID faculty because when we joined, uh, we actually had to take online classes. Some of us were very new to this profession, and we used to miss the crowded classrooms. Then then we also kind of in Delhi, for example, roads were really empty, and this was something was is a unique sight you don't see in major cities. And then, of course, there were other activities which we were missing, and I particularly give this example of a crowded swimming pool because I'll just come back to it related to the talk. Also, we were asked to avoid crowded places because if somebody sneezes, this was what used to happen. Everybody just runs away and kind of leave them alone. So why am I showing you this? Because while the world was getting worried about crowded places and were moving, scared, these crowding was intimidating us, our group was actually interested in understanding the effects of such an environment in a living cell. So this is a picture of a living cell, a cartoon picture, and this is what David Goodsell actually draws it. So a living cell environment actually is made up of large macromolecules and high concentration of small co-solutes or co-solvents and even ions. So we were interested to do molecular dynamics simulations and understand what is the physics behind these effects, what are the consequences of such an environment on biological processes, and use that in material design. So this is the title of my talk. We are particularly interested in self-assembly processes and to understand if we can use macromolecular crowding or this crowded environment as a tool to design nanomaterials. So as I said, a living cell can be called like a crowded swimming pool or a crowded soup, where you have lots of these large-sized organelles, biomolecules, you have sugars, you have also less free water. So the water inside a living cell is not same as what you would see in a dilute aqueous solution. So what does that lead to? That leads to reduced dynamics of these molecules. It enhances intermolecular interactions, like in a crowded swimming pool, people are very close to each other, so there will be steric effects. Then there are volume exclusion effects because of which the other molecules or macromolecules cannot occupy space occupied by certain structures or organelles. And then, as I said, water properties tend to differ. Now the question is, how do these effects actually affect very significant biological self-assemblies that we all know happen inside this environment? So these are the problems people have been interested to understand, and also lately, liquid-liquid phase separation, as Professor Arun also mentioned, is coming up, and people are getting interested to understand this process. So all this happens inside this crowded milieu, and we are interested to ask these questions like, how do these properties of a cellular environment affect these self-assembly processes? So the idea is that if we can understand the thermodynamic driving forces, this is a picture of self-assembled endoplasmic reticulum that's in the ribosome. Sorry, it's a ribosome and the endoplasmic reticulum of the cell. And this is a self-assembly of gold nanorods. These are electron microscope pictures. And the idea is, if we understand the physics and thermodynamic driving forces, can we use such a crowded environment to reversibly tune self-assemblies in nanomaterials, because biofunctionalized nanomaterials, which have applications in biosensors, drug target deliveries, and all those kind of bio applications, because that gives us a reversible tool without destroying the chemical nature of the self-assembling system. So I will revisit a very famous theory which was given by Asakura and Osawa in 1954 to understand colloidal aggregation in a pool of polymer solution. So they had given this theory, and it was found that if you add colloidal macromolecules or colloidal particles inside a crowded solution of polymers, they actually tend to aggregate. So they describe this aggregation or attraction caused by certain forces which are induced by these large size macromolecules in the solution because of differences in the osmotic pressure in the region between the colloids and in the second phase of the solution in the polymers. And they actually came up with the free energy binding expression, which is approximately or is proportional to the ratio of just the size of the colloidal particle and the Crowder or the polymer in solution. So why this is important is because it kinds of, when the big size colloid come together, they are occupying a lot of space or volume in the solution. So when they come together, they release these Crowders and water in the bulk, and that leads to like a depletion of certain molecules from the solvation shell. And that's an entropically favorable process because that results in increase in translational entropy of the depleted molecules. So this effect has been known for long, and these are called depletion forces also. But what is important is the approximation that the Crowders or the polymers have no interaction, attractive interaction with the colloids. They are there just because of they are inducing only size effects. That's what, and they are like hard spheres in the solution. So these approximations are, they could explain many such colloidal aggregation processes over the years, but then also the view that such a crowded environment will always lead to this aggregation. So this was kind of the view that has been accepted over the years, that if you have a crowded environment, it will always lead to folding of biomolecules, compaction in polymers, or aggregation in colloids. So the question that we ask is now that in a living cell, we know that these macromolecules are charged. You have sugars, you have lipids, you have proteins, DNA, etc., ions. So we cannot really have this approximation, we cannot simplify the picture saying that the crowded environment will always have only size effects. It will have no attractive intermolecular interaction with the macromolecule or biomolecule in question that we are trying to study. So then it's more like packing a lot of people in a room and assuming that they will not interact with each other via any other interaction other than the size that they are or the volume that they are occupying in that room. So these are the major 4 questions we ask now. We try to revisit this traditional view of depletion attraction, and we ask that, does crowded environment always induce collapse in macromolecules or cause folding in proteins, for example? Can the macromolecular crowding effects always be attributed to size effects? What is the role of attractions, intermolecular attractions in the system? And if you're using molecular simulations as a tool, like Professor Arun also mentioned, that our current force field that we use usually for these for modeling biomolecules or macromolecules, are they sufficiently accurate enough to represent such an environment? So this is what we ask. And in our preliminary work over the last 2 years, and some other studies have shown that this is kind of the new view, where we are highlighting the role of attractive interaction. So here we did molecular dynamic simulation of a very simple Lennard-Jones 32 bead polymer, which is known to actually exist in 2 conformations in water. Depending upon the temperature or the chemical constituents like osmolytes, if you add, this equilibrium can shift to left or right. So what we did was we actually studied the conformational properties of this polymer in a crowded environment of a tripeptide, trialanine. And we modeled this Crowder. We took a very high packing fraction of trialanine, and we modeled trialanine atomistically. So you can see this is LJ beads, so there are no charges. This polymer can only interact via van der Waals interaction with the solvent and the Crowders. But in this case, what we did was we tuned the polymer and Crowder interaction strength. So these are still van der Waals attractive interactions, dispersive interactions, but we made them strong in one case and we made them weak in the other case. So what we found was, when there are weak polymer Crowder interaction, the polymer likes to be always in the collapse state as you increase the packing fraction of the trialanine Crowders. And so this is the free energy of collapse, and this is the radius of gyration of the polymer, and you can see it goes more and more negative as the Crowder packing fraction is increased. But in the other case, we now make the attractive or dispersive interactions slightly stronger. There is no electrostatic interaction between the polymer and the surroundings. So we see there's a non-monotonic change in the collapse free energy. At very low concentrations of the Crowders, there is collapse state is preferred. But as you actually go in the crowded regime, the polymer unfolds. So this was something interesting because, as I told you, this would be the traditional view that if you put this polymer in a crowded environment, it should always favor collapse. But then we found this is interesting. And what we also found was, in this case, when unfolding happens, there is preferential adsorption of these crowders on the polymer chain. But when this collapse happens at high concentration, there is depletion, the traditional depletion effect that we usually see. So this was something interesting, and we could see that the role of attractive interactions is something crucial in understanding the equilibria, conformational equilibria in such responsive polymers. So then now we are studying another system where we would like to understand the self-assembly in kind of fibril formation. So this is a cationic dyestuff. It's pseudo-isocyanine chloride, and it's in a very interesting dyestuff. It's a small molecule, but it is known that when you add a large concentration of this dye in water at a particular concentration onwards, or if you change the temperature, these dyes actually form fibrils. They form stacked structures, like beautiful stacked structures, which is similar to what you can also find in some certain kind of proteins called intrinsically disordered proteins that form amyloids. So also interesting is that if you look at the structure of this dye, it is cationic, then it has these hydrophobic tails, side chains, and also it has a lot of pi-pi interactions. So this kind of also has a lot of features which you would find in a small peptide. And that's how it becomes interesting to if we can understand how this stacking of these dye molecules happen. That will be interesting to understand how actually, if we can correlate this to the understanding of amyloid formation. Recently, it was found that the experimental groups, they actually injected these dyes in a cell where they had increased the salt concentration. So osmotic stress was very high in the cell, because of which the crowding environment, like the crowding effects, increased. And they found that the moment they add these dyes inside such a cell, these dye give a very good fluorescent signal. So these dyes can actually act as molecular sensors for sensing crowding in the cells. And this is something very interesting because many diseases like cancer, in some of the cancers, cells actually solidify, and the diseased cells. And this dye can actually act like a very good sensor to sense such environments. So we want to understand that what is the effect of this crowded environment on the aggregation of these dyes. So there was another experiment where they found they actually, so this is the two kinds of aggregates these dyes can form. So H aggregate is when the dyes are just face-to-face stacked, and J aggregate is when the dyes are slipped. So you can see the alternate arrangement like this. But what is found interesting is that the UV-vis spectroscopy have shown that the J aggregate actually absorbs at a very sharp, high wavelength, whereas the H aggregate, they are called like a hypsochromic shift is there, and they absorb at a low wavelength. So it becomes interesting to see if you add Ficol 400, which is a high molecular weight sugar, in such a solution, there immediately J aggregates are formed, and you see a very sharp absorption signal at 575 nanometer. But if you add very commonly used Crowder, a neutral polymer, polyethylene glycol, then no J aggregates are formed. So itself, this itself is showing that the chemical nature of Crowder is important. In one case, you have sugars which have all partial charges, a lot of OH groups, and that is actually inducing the aggregation, whereas a neutral polymer, polyethylene glycol, is not. So now the question arises, can we look at it microscopically and understand what's actually happening? So we try to understand that, and we do molecular dynamics simulations. So the challenge here was to actually get the aggregates. So we are trying to form a small stack of these H aggregates. These are 12 dye molecules only, and you can see they are very unstable in water. They don't really stay in that conformation. So we had to actually supply certain position restraints. So students who are working in molecular dynamics would understand. So we apply certain force constant of harmonic forces to let them stay there, and then try to understand the structural properties. So here, these are just RDFs of these dye molecules interacting with one another. And this is in pure water solution, and this is the crowded solution of ethylene glycol. We started with ethylene glycol, and we can see that you see this crystalline structure of the dye in the RDF in both the solutions. right? But now if you go to J aggregate, you see, look at the RDF of the ethylene glycol solution, the propensity to form a J aggregate immediately decreases as compared to H aggregate, which was similar in pure water. Here we see the differences. So this was the first indication that the ethylene glycol, which is the family of monomer of polyethylene glycol, is not supporting or stabilizing this J aggregate. Then we try to understand the binding free energies of these oligomers to understand if we start forming high order stacks, how the binding free energy would change. So here we did umbrella sampling simulations, MD simulations, and you can see that if when you go from a dimer to trimer to tetramer to pentamer, from dimer to tetramer, stability in pure water is increasing, but as soon as we go to the fifth one, the pentamer, somehow the stability decreases. And then similarly in aqueous ethylene glycol, this is a crowded packing fraction or concentration of ethylene glycol. And here we see, although dimer to trimer, there is high binding, there's a high stability, but on forming another higher stack, the stability is not as much as you saw in case of pure water. So ethylene glycol here we can see is kind of destabilizing these oligomers. So this is nothing but now the binding free energy from the PMF, the potential of mean force plots. And you can see that the dimer formation, this is H dimer formation, which we saw in the spectrum of experiments also. There, both pure water and the solution of ethylene glycol support dimer formation. But as we increase the stack size, in pure water tetramer is still stable, but ethylene glycol has lower free energy of binding, higher free energy of binding as compared to that in pure water solution. So this already is supporting the experimental observations that ethylene glycol as a crowder does not stabilize even the oligomeric structures. And these are just H aggregates. We are still exploring now the J aggregates, how ethylene glycol would perform as a crowder for J aggregate formation. So to understand this further, to understand whether depletion is happening or adsorption is happening, how ethylene glycol is actually destabilizing these oligomers, we can actually go further and compute the one parameter called preferential binding coefficient. So if you consider this equilibrium reaction between the monomer and the dimer, the change in the chemical potential is proportional to this equilibrium constant, and the partial differential of this chemical potential change as with the activity of the crowder will actually give you the change in the preferential binding of crowder over dimer and the monomer. So what this means is that although I'm calling this binding preferential coefficient, so when something is binding, it necessarily is not an energetic effect. It's a free energy effect because here we have the change in the chemical potential. So this is something what experimentalists also do in the dialysis experiment. You have a semi-permeable membrane. On one side, you have, say, protein and a denaturant, and on the other side, you have the denaturant, and you keep on changing you let the denaturant flow through the semi-permeable membrane and measure the change in the concentration of the denaturant with the concentration of protein. And that's what this preferential binding coefficient is. So this gives us an idea whether the equilibrium, will shift to left or right if we can compute this quantity from simulations. And I will not show the details of this analysis. So we can further dissect this chemical potential change into entropic and energetic contributions based on Widom potential distribution theorem, which is nothing but expressing this chemical potential change in terms of attractive solute solvent energy and solute solvent entropy change. So that results I will not show because of limitation of time, but I would like to show these results where we could compute the preferential binding coefficient, and we can see that if gamma is positive, it means that the crowders are actually adsorbing on the surface of dyes. But if it is negative, it means they are depleted from the surface. So this shows that ethylene glycol is actually adsorbing on the surface and destabilizing these oligomers. So this is something what we found. It's this often is something like this picture where all the crowders, they actually come on the, sit on the surface and destabilize the conformation, folded conformation of the macromolecule. So the interplay of energy and entropy, we did some analysis, but that also showed that both entropic and energy, there's a delicate balance between the two, which actually determines whether these stacks of oligomers will be formed or not. So what's the take-home message? We today looked at the new view of crowding effects, and we saw that the crowded environment may not necessarily always lead to collapse of macromolecules. It can lead to unfolding of proteins as well, as some studies have shown. We also saw that the relative strength of these attractive intermolecular interactions between the crowder and the polymer or your dyes in question, or the aggregates in question, that is very important. That plays a very important role in determining whether aggregation will happen or not. And there is a case where we saw that the crowders had actually adsorbed on the surface and unfolded the polymer and also destabilized the aggregates. So this depletion and adsorption is a very important factor that determines where the equilibrium will be shifted. So that's all from my side today. So then I would also like to show some other work we are doing in actually accurate modeling of the crowded environment, because what we see is that we took very simple amino acids, deuterionic forms, and we ran simulation at say 3 molar, which is packing fraction of about approximately 20%. And we find that amber, charm, these kind of force fields, they actually show aggregation. The amino acids tend to aggregate. So there was a study where they have actually tuned the termini Leonard-Jones size parameter and the sigma ij, and that they just add delta sigma to it, and that actually leads to such a nice de-aggregated or unaggregated system. So we have done some work in which we present 5 amino acids and how the osmotic, we have computed osmotic coefficients using the simulations, and this is gray one is the experiment. You can see when you use original force field for different amino acids, there's a large difference between experiment and simulated values. But after using this reparameterization, there is a very huge, there's a large improvement in the values of osmotic coefficients. So this is one work that we are trying to establish. And then we are also using these ideas of actually functionalizing gold nanoparticles with single-stranded DNA and design self-assemblies with different concentrations of crowded environment. So I thank you all for your patience, and my group members. I have 2 PhD students who have contributed to this work, and some work I had done at IIT Kharagpur, which I showed today, and some project interns, and I am collaborating with Professor Klaus on the dye work. So, and also I thank the funding agencies, and thank you all. I can take up some questions.
Thank you, Divya. It was a really nice talk, and thanks also